So my name is Paul Danis with the consortium. Welcome to the 5th installment of Proteoform Thursday. We're lucky today to have Kyle Brown speak to us about some of his recent work on membrane proteins. Should be fascinating stuff. Let me give you a quick intro on the consortium, a couple of bits of news I want to share, and then we'll get going here. So the consortium is two fronts we operate on. One is building the community, which is doing events such as this here, as well as educational programs. The other is trying to guide the technology, and I'll have a bit on that in a second. We'll try to develop protocols, expand databases, and things like that. So those are the two fronts we work on. An example is last week, we just launched a new initiative to try to understand CEMS. There's a lot of challenges around protein separation, and CE is a really valuable tool for that, but it's challenging. So Liang Liang's son, Alexander Ivanov, and Kevin Jost are leading this new initiative to try to first assess the state of the art around CEMS and CEMS-MS for doing top-down work, and then running an interlab study where they're going to try to see where is the best practices, where are the things that are working. And based on that, they'll develop and publish guidelines for people who are looking to get into CEMS or refine their processes and to try to expand the ability of people to do this kind of work. The other thing we ask people when they registered, like how important would be a proteoform database to your work? And this is, again, there's a bias here because this is people that registered for this. There's over 100 people here. So, but a good portion of them said this is really, really important thing to have. We do have kind of an alpha version up right now, and we're looking to expand that. But it's important to understand this could be important for a lot of people's work. Some people would use it occasionally, and some folks don't need it at all, which makes sense. So one thing we'll probably be digging in for the folks that definitely need this kind of database, we'll probably try to dig in a little more, understand what specifically would be kind of the uses you would have or the applications that would be really beneficial so we can help design this thing properly going forward. So we'll be talking with you more about that in the future and looking for the community to help guide some of that work. There's also a new course for top-down proteomics that'll be this fall at ASMS. You can register at ASMS. As you can see, there's a STEAM panel be presenting some of the finest work there. And so we look forward to that. So it's going to cover the key areas if you want to get into it, if you have colleagues who want to get into it, or if you're looking to really understand areas that you maybe need a little help in. This is a great crew, and I'm sure there'll be lots of good discussions besides what's on just the syllabus here. So go to ASMS and you can sign up for that. So next month, Frederick Lermite is going to be speaking to us about some of his work in native top-down that he's done. That should be fascinating. I know Frederick's got a lot of passion around this area, and he's also leading our initiative in native MS as well. So tune in for that. There'll be more details coming over the next week or so. So if you're not a member yet, please go ahead and join. It's free to join the website. You can go to topdownproteomics.org, or if you don't want to type that much, just proteoforms.org. We'll get you there as well. And finally, thank you to our sponsors, Thermo Fisher, Brooker, and SciEx, as well as some of our supporters, Agilent and Pfizer, who are enabling us to do these kinds of programs. And we really appreciate their support for the consortium. So without further ado, I'll turn it over to Kyle, and he'll speak to us about some of his work on membrane proteins. So Kyle, go ahead, take it away.
There we go. All right, come on. All right, can you see my slides, or am I no longer sharing? I think my PowerPoint just crashed.
Yeah, we're not seeing it right now, Kyle.
Okay, 1 second.
That's good.
All right, now we're rolling. All right, so.
And Kyle, if you could get a little close to the microphone, you're a little bit soft.
Okay.
Thank you.
Is that a little better?
Yeah.
All right. So sorry for the technical challenges there. So with that, the title of my talk is Novel Strategies to Characterize Membrane Proteoforms Using Top-Down Proteomics. And I'd first really like to thank the organizers of this Proteoform Thursday for giving me the opportunity to talk. It's been such a fun seminar series getting to hear such amazing work in the field. And I'm glad I could be a part of it. So a little bit about me before we get into it. So I got my undergraduate degree at the University of North Carolina at Chapel Hill in chemistry and mathematics, where I was introduced to research working for Professor Ken Hardin in the Department of Pharmacology, working on purinergic receptor signaling. And then I was very fortunate to have the opportunities in the summer to go back to my home and work at Vanderbilt with Professor Craig Lindsley in medicinal chemistry. And then after graduation, I had a brief summer intern at Merck where I worked on separation of biotherapeutics, which led nicely into my PhD work with Professor Inga at the University of Wisconsin, working on top-down proteomics of membrane proteins, among other things. And then currently I'm a postdoc in the lab of Professor Sam Gelman working on some chemical biology projects relating to G-coupled protein receptors. And so today I'm really going to focus on my PhD work in Professor Gug group. And I was really lucky to work on a number of different projects. And kind of the central theme of my work was to develop methods to characterize challenging subproteomes. But first, to kind of start with the basics of why we characterize proteins, we know that as biological information flows from DNA to RNA to proteins, that variations can arise as proteins are produced from alternative splicing events and particularly from post-translational modifications. The result being that from a single gene, you can have multiple different molecular forms of a protein that we call proteoforms. And these are really the functional units of the cells that really carry out the features that we depend on for life. And so there are two general approaches to characterizing proteins using mass spectrometry. So there's the conventional bottom-up proteomics approach, wherein proteins are digested, typically using an enzyme, and then sequenced using mass spectrometry. And this affords generally pretty high proteome coverage, so you can identify upwards of 10,000 proteins or more. It's highly quantitative and well-developed, but you do not get proteoform information using this technique, and I'll touch on that a little more specifically. If you instead sequence intact proteins, you do at least currently lose a little bit generally in terms of the proteome form, the proteome coverage. However, it does allow for very rich information regarding the post-translational modifications in the isoforms that are present within a cell. And it's somewhat less developed in the more conventional bottom-up approach. But what's really key about looking at intact proteins is that you can really precisely define what proteoforms are present. And when you digest proteins, you can lose information about what proteoforms are actually present and again, functioning. And this is very relevant in the context of disease research, where a proteoform change can actually result from disease progression. So as we can see here, there is a change from the phosphorylated forms of this protein down to the unmodified form of this protein as congenital heart failure increases. So being able to detect and analyze proteoforms can be critical for disease detection early on as you can monitor proteoform changes. Understanding how proteoforms change in the context of disease really gives us insight into disease mechanisms. And of course, when we understand how proteoforms are changing, we can then develop new treatments around preventing those changes from occurring, or at the very least reversing them. And then because proteoforms are such a functional unit within the cells, this is really an opportunity for more personalized medicine. So developing therapies or giving therapies based on proteoform level changes versus kind of disease, maybe more genetic changes, which could be broad. However, there's a number of challenges still facing implementing top-down proteomics, particularly on a global scale. So some proteins are very difficult to solubilize. And the proteome is very complex, and intact protein separation is much more analytically challenging when compared to the separation of peptides, which are a little more friendly and easier to handle. The proteome has a very large dynamic range, so that there's some proteins that are very high in abundance and some proteins that are very low in abundance. And this can make it very challenging to kind of study all of the proteins, which is kind of the ultimate goal. And probably the most severe or our greatest challenge is this phenomenon wherein as your molecular weight of your analytes increase, you have a decrease in your signal to noise ratio because you have a greater number of charge states and a greater number of isotopes, which really makes it challenging to study high molecular weight species, especially because you have a much greater signal to noise ratio for the low molecular weight species. So that makes it difficult to study these proteins. And all of these challenges are kind of heightened for one category of proteins, which are membrane proteins. So these are proteins that are found in the lipid bilayers of cells or interact with them. And they constitute about 1/3 of protein encoding genes within cells. And they're involved in many important physiological processes. But probably most notably, they account for a disproportionate number of drug targets. So I've seen as high as about 60% of drugs on the market target membrane proteins, in particular G-coupled protein receptors. So from a therapeutic standpoint, they're very important. As I kind of indicated before, a lot of their role is to relay signals from the outside of cells to the inside of cells, or to transport molecules into and out of the cells. And these have obviously physiological consequences downstream. And of course, in the context of disease, we all know that membrane proteins play a crucial role, for example, in the entry of viral capsids into the cell begins at the ACE2 receptor for coronavirus, for example. So there's a lot of work in terms of targeting this class of proteins for next generation therapeutics. So why they're so challenging to study, particularly in the context of top-down proteomics, is if we map the molecular weight of human transmembrane proteins, we see that the average molecular weight is about 60 kilodaltons, which for top-down, that's relatively large. Moreover, if we look at the abundance of these proteins in comparison to other protein classes, we see that they're expressed at significantly lower levels than other classes of proteins. So you just have less copies of these proteins per cell, which makes them obviously more difficult and requires greater sensitivity or greater enrichment. And probably most notably, membrane proteins are extremely hydrophobic. So they're relatively insoluble in the aqueous buffers that we traditionally like to use for mass spectrometry and for biology. And to circumnavigate this problem of insolubility, we typically use, in biology, we rely on surfactants. So these are a class of amphiphilic molecules that are generally characterized by their head groups. So we think of anionic and cationic surfactants, which are charged, zwitterionic, which have charge but are net neutral, and non-ionic. And these are kind of characterized by these charged ones are the most denaturing and kind of the harshest, whereas the non-ionic surfactants are kind of the most mild. And the properties that we really take advantage of are they form micelles, which protect especially hydrophobic proteins from water and keep them soluble. And they disassociate the membranes of cells, so it allows for efficient protein extraction. However, despite the fact that these are widely used in biology, unfortunately, they generally have a very deleterious effect on electrospray ionization mass spectrometry. So if we look at one of the most powerful solubilization agents in SDS, we see that at even very low concentrations, it almost completely inhibits mass spectrometry signal. So despite the fact that these are so widely used and really powerful agents for protein solubility, they inhibit our ability to do mass spectrometry. So to circumnavigate this, one approach is to that individuals have taken is to put a cleavable bond in between the hydrophobic and hydrophilic portions of these, such that you can degrade the surfactant and more easily remove it and allow for mass spectrometry analysis. And this has been conventionally done using acid cleavable motifs because it is very orthogonal to the general, to the kind of traditional proteomic workflows, which are done generally in positive mode, so acidic conditions. However, as I'll get to in a little bit, these acid cleavable surfactants, which have been used in bottom-up proteomics, are really incompatible with top-down proteomics. So we decided to take a different route. Instead of using acid motifs, we used photocleavable motifs. So we designed 3 classes of surfactants with different photocleavable linkers in between the head groups and the tail groups. And then we synthesized different chain lengths to balance the hydrophobicity of these surfactants. And so after synthesizing these surfactants, we first characterized whether they were water soluble. This is kind of a common stumbling point for surfactant development, is actually in some cases they're too hydrophobic and they're just not soluble in water and therefore not really usable. So we could kind of discount a number of surfactants based on that. And then we tested whether they effectively extracted protein, so whether they increased protein solubility. And just using these 2 simple characteristics, we pretty much discounted a lot of different surfactants and really found only one surfactant that afforded both of these qualities. And that was one with a azobenzene linker and a 6-carbon chain. So what this workflow kind of looks like is we now have this photocleavable surfactant that would allow us to solubilize proteins and extract proteins. And then when we treat that with UV light, it degrades into more innocuous byproducts that don't have the surface activity of the original surfactant and are more easily removed. And we can see the maximal wavelength of this is around 305 nanometers. So that's the light, generally light within this region is required for the cleavage. And the benefit of the photocleavable route in particular, as compared to the acid cleavable route, is we can see this is done on a really fast time scale. So it really takes only a couple of minutes to degrade this surfactant, which is really important. And so the first thing we wanted to do is look at how well our surfactant performed for protein extraction. So we homogenized cardiac tissue, in this case, in aqueous buffer, and then to remove the soluble proteins. So these are proteins that you can really solubilize in water, and then centrifuged it down to give us this insoluble pellet, and then treated that pellet with different surfactant conditions, and then used SDS page analysis to kind of look at the extraction efficiency. And not surprisingly, the anionic surfactants performed really well. That's why we conventionally use these for extracting proteins. So we see the acid labile surfactants as well as commonly used SDS and our photocleavable surfactants really performed very well. Whereas the more mild non-ionic surfactants were really not as efficient at extracting protein in comparison. So from there, we wanted to look at the compatibility of these surfactants for top-down analysis. And so after cleaving the acid cleavable surfactants and then directly analyzing them using mass spectrometry, we can see very severe signal suppression when compared to the control. And of course, when we analyze anything in SDS, it completely removes our signal. On the other hand, if we look at the non-ionic surfactants in our photocleavable surfactants, we see a relatively good signal when compared to the no surfactant conditions. So these are much more mass spec friendly when compared to the acid cleavable surfactants. And I want to draw your attention to a few kind of key components that are key considerations. So we use organic solvent in our matrix, not only because it helps for effective electrospray ionization, but also later on, it will help retain the solubility of hydrophobic species. So once we remove the surfactant, we need something that will help the surfactant stay in solution. And organic solvent is particularly good for hydrophobic species. In addition, we include TSEP, which acts as a radical scavenger and really prevents any protein oxidation that might occur during photoirradiation. So combining these 2 experiments, we see that when we look at top-down compatibility and protein solubility, really only we found that our photocleavable surfactant really kind of gave us the best of both worlds. So when looking at both characteristics, this one was the best. So then we kind of moved forward with this to use it in order to study cardiac proteoforms. So again, we took cardiac tissue and depleted using heaps extraction and then ultracentrifugation, and then treated this insoluble pellet with our surfactant and performed a workup. And when we looked at the proteoforms that we were able to identify, we see that many of them are coming from the mitochondria. For example, this string analysis over here shows proteins that are involved in the electron transport chain. And this is not surprising given the requirements of cardiac cells to produce a lot of ATP. So these are generally abundant proteins. So looking at some of the mass spec data or the top-down data that we really can get out of this, we were able to identify phospholambin, which is a key cardiac regulatory protein that I'll talk about more in a little bit. We were able to identify this receptor expressing enhancing protein 5, which has 2 transmembrane domains and has recently been implicated are being involved in ischemia. And then we were able to identify every subunit of the ATP synthase complex. And we were really encouraged by these results because not only were we able to see kind of the soluble region that sits kind of in the matrix, but we were also able to identify all the subunits in the hydrophobic portion. So the ones that are actually in the membrane. And these are ones that would really require surfactant technology in order to analyze. So one of the proteins, as I mentioned before, that was really interesting in terms of cardiac biology is phospholambin. So this protein is a key regulator of calcium into and out of the sarcoplasmic reticulum, which dictates contraction. And we were able to localize a lipid modification in the transmembrane domain on the cysteine residue. So this is just demonstrating how this surfactant technology enables the study and characterization of proteoforms that would generally be difficult to analyze. In further work on this protein, we've been able to identify a number of different proteoforms, including the singly and doubly phosphorylated, as well as the palmitoylated and even the palmitoylated phosphorylated form. And this really speaks to some of the advantages of top-down proteomics because there's so many different forms of this protein, and there's a lot of potential regulation of this in cardiac cells, which we are currently looking into. But being able to precisely define what proteoforms are present and how they are functioning is really something that top-down can really aid in. And so we've expanded this surfactant technology for a number of different applications. First, we demonstrated the use of it for high throughput bottom-up proteomics as well as top-down. So we really have this integrated strategy wherein we can take a sample and use this photocleavable surfactant to extract proteins and then kind of take it and run both bottom up and top down on the same sample to kind of maximize the amount of information we get from a sample. And this method was refined a lot more by Timothy Abello in our group, wherein he really demonstrated how you could scale this down to really small amounts of tissue, so only 1 microgram of tissue. You could perform very fast digestion conditions, about 30 minutes, and then do a quick cleanup. And then using the timsTOF, you could actually identify more than 5,000 proteins, I believe. So it really enables very efficient bottom up. And we've also demonstrated the use of this technology for looking at extracellular matrix proteins using bottom up. So there's a number of different applications that we're showing this photocleavable surfactant has some utility in. So in summary, just to kind of summarize what we've seen, so we developed a photocleavable surfactant that was top-down friendly. We demonstrated that it effectively solubilized proteins, including membrane proteins. And kind of most notably, it enabled high throughput sample preparation. So that's really where its strength lies. So some of the remaining challenges that came out of this study was that we found that only about 28% of the proteins we identified were integral membrane proteins. So we saw a lot of other protein classes, including nuclear proteins, structural proteins, extracellular proteins. And that's not surprising because these classes of proteins aren't necessarily soluble in the aqueous buffer. So depleting them just by homogenizing them in an aqueous buffer and then treating them with a really powerful surfactant, it's not surprising that we saw these other protein classes, but it does mean that we need more effective enrichment if we really want to target the membrane subproteome. And then we also, most of the proteins that we detected or identified were low in molecular weight, which is again, a general problem with top-down proteomics, especially since we were only using one dimension of separation, just reverse phase liquid chromatography. So to address the challenge of enriching membrane proteins, we decided to take a different approach than just depletion by homogenization in aqueous buffer. So instead, we wanted to take advantage of the unique properties of this surfactant Triton X14. And what's kind of interesting about this surfactant is at cold temperatures, it behaves just like any other surfactant. It will lyse cells, solubilize proteins. But as you warm it to room temperature, it forms two distinct layers, a surfactant layer and a water layer. And what's important about this is back a while ago in the 80s, they found that membrane proteins were really sequestered in the surfactant-rich layer, whereas water-soluble proteins were found in the water layer. So what this enabled you to do is lyse cells and then just bring the sample to warmer temperatures and form this two-layer, and then just pipette the hydrophilic proteins away. So you have this really membrane-enriched sample that's very easy to acquire. However, because we're using Triton, which is not as mass spec friendly and really inhibits signal, we do have to perform precipitation to remove the surfactant. And so that afforded really effective membrane protein enrichment, as I'll discuss in a little bit. But we also wanted to enable the detection and analysis of larger proteoforms. So we needed a size-based separation that would enable that analysis in addition to our reverse phase liquid chromatography. So in the Go group, we really like SEC. And one of the reasons it's a relatively fast analysis. So in this case, we're doing offline SEC, and it takes about 20 minutes for a run. And if you want to pool a couple of fractions together, then it takes 40 minutes or an hour. But we can also use mass spec friendly solvent systems. So in this case, we're using isopropanol, which again helps us maintain solubility of hydrophobic proteins along with formic acid. And so if we look at the SDS page analysis of our SEC fractionation, we can see a pretty good size distribution of our proteins. And again, if we look at the average molecular weight of the proteins we detected using reverse phase LCMS, we see that it increases essentially with our molecular weight fractions. But not surprisingly, if we look at the number of proteoforms we detect, we're still detecting a lot more proteoforms in our smaller fractions. And right here on the right, we can see representative examples of some of the proteins we were able to detect using this SCC reverse phase LCMS. So we get a good distribution of masses ranging from about 115 to about 7 kilodaltons. But kind of most notably of taking this approach is we were able to detect really large proteins for the first time in comparison to not using a size-based separation. So that was one of the really powerful advantages of using SEC, incorporating that into our workflow. Moreover, if we looked at the proteins we identified, about 70% of them were membrane proteins. So again, we have a really rich membrane protein fraction, and 54% of them were integral membrane proteins. So what this gives us is it gives us a really nice handle to develop new analytical strategies and new top-down methods aiming to characterize membrane proteins. So for example, we looked at our chromatography, our reverse phase separation, and we looked at specific proteins and the number of transmembrane domains that they had. And kind of interestingly, we saw that the number of transmembrane domains really was not a good indication of retention. So we have proteins with relatively small number of transmembrane domains that are actually really hydrophobic and elute really late. And we have other ones that have very high numbers of transmembrane domains that actually elute relatively early. So on the other hand, if we use a gravy score analysis, which is just the measure of the hydrophobicity based on the amino acid sequence, this gave us a much better prediction of retention as compared to transmembrane domains. So by being able to look at a more membrane enriched fraction, we can really tease out some of this interesting information that will help. Potentially, we can use this type of information to develop better chromatography strategies or better data analysis strategies based on retention and retention prediction and things like that. And so that's what this method really affords. We were also able to localize post-translational modifications of these integral membrane proteins, which was really obviously what kind of the ultimate goal. And we tried this with 2 different systems, and we were able to identify a number of integral membrane proteins from a number of different organelles. So we didn't necessarily have to enrich for one organelle. We got proteins that were nicely distributed across them. And some of the proteins that were not necessarily integral membrane proteins were membrane associated proteins. So those are those that belong to complexes or form protein-protein interactions. And it's not surprising that we saw those given that the extraction is relatively mild and native until we get to the SEC. So to summarize this part, the cloud point extraction really afforded effective enrichment of membrane proteins up to about 70%. Our SEC method was relatively high throughput, and we got pretty good size-based fractionation, being able to detect proteins as high as 115 kilodaltons and as low as about 6. And we identified membrane proteins with transmembrane domains from as much as 19 from 2 different systems. However, I will note that the cleanup necessary for removing the triton and of course the offline separation resulted in lower throughput. So that's kind of the weakness of this method in comparison to our first one. So the first 2 projects I've really discussed are kind of targeted towards global membrane protein analysis. So really trying to characterize as many proteoforms as possible from a given sample. But with the remaining time, I'd like to briefly touch on our work on targeted membrane proteomics. So there's some cases where we really want to look at and deeply characterize a single protein. And in this case, we were looking at the inward rectifying potassium channel, KIR 2.1, which is important for maintaining resting potential of membranes. And significantly, mutations of this protein are implicated in several sudden death syndromes, and they're often triggered by beta adrenergic tones, which is to say that if you know anything about the adrenergic pathway, generally stimulation of that receptor results in downstream kinase activation. So when we think of kinases being activated, that leads us to believe that phosphorylation appears to be a critical regulator of this protein. And there's some indirect evidence that phosphorylation plays a role and has a function, and some indirect evidence on maybe some sites, but there's not a clear understanding of where this protein is phosphorylated. So we set out to use proteomics to discover where this protein was phosphorylated. And so we really wanted to study the functional channel. So we express this in HEC293 cells, so mammalian cells. So often membrane protein studies are done in bacteria cells, or they're done in insect cells where you don't have necessarily the interacting partners or the relevant post-translational modification machinery. And so we really wanted our method to look at it in mammalian cells. And we used electrophysiology to characterize this receptor to make sure that it was functional. And then we performed affinity purification to purify it and enrich it. And then we used both top-down proteomics and bottom-up proteomics to characterize it. So looking at the kind of results that we got from our top-down analysis of this, so just looking at the basal level, after kind of developing a top-down method to analyze this protein, when we looked at the basal level, we saw just a very small amount of phosphorylation. we were able to get pretty good mass accuracy using a QTOF instrument. And when we did online CID, we noticed that a lot of our fragmentation was kind of in the N and C terminus region of this protein. And additionally, we did, our collaborators did whole branch clap analysis with and without the tag, because in some cases a tag can inhibit protein function. And in both cases, we saw a typical N-shaped outward current. And the maximal signal of that was around -50 millivolts. So we were pretty satisfied that we were working with a functional channel. So with that, because again, some of the sudden death syndromes are associated with beta adrenergic stimulation, we looked at this protein both with and without pKa stimulation. And not surprisingly, when we stimulated the cells, we saw a rise in the monophosphorylated and bisphosphorylated proteoforms. And using online CID, we were able to localize a phosphorylation at serine 425. And that's just based on without the MEC tag. So that would be the kind of endogenous location. So naturally, we wanted to kind of characterize the phosphorylations further. So we mutated this serine to alanine, and then again stimulated the cells. And what we found was actually surprising. So we thought by mutating this site, we would really see a phosphorylation decrease. But after mutation, we actually still see a fair amount of the monophosphorylated and bisphosphorylated proteoforms. And what we saw when we did online LCMS of this was that we saw phosphorylations now at the N terminus of the protein rather than the C terminus, where we identified the original phosphorylation site. So there's this kind of what appears to be an interesting switch in phosphorylation upon mutation. And so moving forward, we wanted to dig even further into the phosphoproteome of this. So we employed bottom-up proteomic analysis. And that enabled us to identify a few more sites internally on the internal part of the protein here. So we identified 3 more sites. And I kind of wanted to draw your attention to even though we have pretty good sequence coverage of this protein, about 84%, we see that the serine that we previously identified as phosphorylated in the wild type cells, we don't get sequence coverage of that. So that's kind of the information that can be lost when you do digestion. So this is why an integrated top-down and bottom-up approach can actually be quite informative. And again, we did the same type of bottom-up analysis on the mutant and verified not only the 13 and 14 phosphorylations, but again, a more internal site. And kind of interestingly, we found that we identified peptides that corresponded both to the monophosphorylated 13 and 14, as well as the bisphosphorylated. So we can see a pretty complex phosphoproteome for this protein. And so overall, this led to 6 novel phosphorylation sites that were identified for this protein, which was really gives us a kind of new areas to explore what are the functional consequences of these phosphorylations, which our methods really helped. So in summary, we developed a top-down proteomic method that allowed us to see the overall, kind of the overview of the proteoforms of functional care R2.1. And our top-down approach enabled identification of phosphorylation sites at the N terminus, as well as those that arose after mutation on the C terminus. Actually, sorry, that should be switched. And then bottom-up proteomic allowed for further mapping of phosphorylation sites for a total of 6 novel site identifications. And one takeaway that I kind of had from this, particularly from a top-down perspective, is there's still room to improve kind of the precise characterization of target membrane proteoforms. So we had trouble, for example, getting data on some of the internal phosphorylation sites using online LCMS. So implementing alternative fragmentation strategies, improving sensitivity are just some of the things that will make this work easier and more informative. And that's some of that work I'm going to be carrying, I've been carrying with me to my postdoc research. Instead of on ion channels, it's been on characterizing GPCRs. So we're developing strategies to better characterize the post-translational modifications of these important proteins, while also integrating some chemical biology and pharmacology to characterize their signaling profiles. So with that, I would like to thank both my PhD and my postdoc mentors, all of the group members over the years that have contributed greatly to this work, our collaborators, Dr. Andrew Alpert, who helped with the SEC, and Dr. Lee Eckert, who we worked with on the KIR 2.1. And of course, the training fellowships that have helped me in the research funding. So with that, I would definitely take any questions that people have.
That's great. Thank you very much, Kyle. That's great stuff. Really appreciate it. I just had if folks have questions, you can either ask them live or drop it into the Q&A if you'd like, and we'll go ahead and process that. I just had a quick question on those photocleavable surfactants. Are those, I assume those are things you guys custom synthesized. How could other folks get their hands on those? Is the synthesis published, or are they being commercialized, or just curious what you think about that?
Yeah, great question. So we did synthesize all of these in-house. I did kind of, especially the work on the azo surfactant was things I synthesized. They're relatively easy to synthesize, at least the azo surfactants. It's a two-step, and it's a pretty good yield on gram scale. And you essentially, it just precipitates at the final step. So there's no need to run columns or anything like that. So we do make it in-house fairly easily, and anybody with some synthetic skills can do it. And there are published, not only our publication, but other previous publications on this synthesis. And then on top of that, we are working with Sigma to commercialize this surfactant to get it as a tool for very broad use. So I hope that answered both those questions.
Yeah.
Great, thanks.
Thank you, Kyle. Actually, just a quick comment. You know, Sigma is supposed to have the product launched back in February, but there's a big delay. So if any of you interested using the azo and do not have synthetic capability, as many analytical chemists, do not have the synthetic capability in the lab. So please just reach out to me and to Kyle, and we should be able to send you some samples for you to test.
Great, thanks, Ing. Tariq has a question.
Yes.
Hey, great talk. Have you tried knocking out any of those phosphorylation or putative phosphorylation sites to see if there's any electrophysiology effects?
Yes, that's a fantastic question. So we knocked out the serine 425 and then did electrophysiology on that. And there was not a statistically significant difference in the output current. So that suggested to us that perhaps the total level of phosphorylation was more important than the site-specific phosphorylation, especially because if you look at the structure, the C terminus and the N terminus both kind of hang out of the cell. But we need to generate more functional data to verify that trend. But that's exactly what we're trying to do now that we've discovered all these sites, is knock them out and then look at the functional consequences downstream, as well as looking at the mutations that we know cause diseases. But as you can see, it might be a little tricky, because as soon as we knocked out one site, new sites appeared. And so you have to generate stable cell lines with single and double mutations. And we have 6 sites, so that's potentially 6 cell lines plus double mutants. So essentially, we kind of, at this point, are kind of closing this story, and our collaborators are really honing in on the functional consequences of these sites, because there's tons of work now to do on that, which is really exciting. But thank you. That was a great question.
Great work. Thanks.
Hey, thanks. I had another quick question on the multidimensional work. You mentioned that the throughput's kind of challenging and things. Do you see any ways to try to automate the multidimensional work to make that a higher throughput process? Or is that always going to be kind of a sneaker net process?
Oh, that's a fantastic question. I think there's a lot of room for automation. I think there's already some great work that's been done by, I know Vicky Wysocki published some online SEC that was mainly for desalting purposes, but I think there's already growing room for multidimensional separations all online, 2D-LC online. So I do think that you could couple online SEC with reverse phase to greatly increase the throughput of these methods. So I definitely see this as a limitation that is going to be solved with new technological developments.
Great, thanks. And we have a comment question from Adnan. He says, it is amazing that your top-down proteomic data contain VDAC proteins.
Oh, yes. That was definitely, we were very, very encouraged by that type of data because it was something that we had really never seen, at least in our hands, had never seen before. And it really speaks to how well the cloud point enriches membrane proteins. So we get this really enriched membrane protein fraction that allows us to see proteins that are generally really challenging to see. So we were very impressed by that enrichment performance. So you were very correct. We were very happy to see those types of proteins.
Hey, Kyle, on slide 24, when you're talking about the cloud point enrichment, you say that there's, additional cleanup steps. Are those cleanup steps the subsequent SEC fractionation, or are you doing more?
So in this case, we do, great question. So I didn't see who said it, but in this case, we used precipitation to remove the bulk of the triton, and then used SEC as an additional cleanup method. So those are the two methods we use, though I will say that SEC alone can remove the most of the surfactant. You just have a lot of surfactant in your LC. So to kind of be a nice lab citizen, we removed a lot of it early on. But yes, the precipitation and the SEC afford great cleanup.
And the precipitation is a methanol chloroform water precipitation, or what are you doing?
Yeah, correct. In this case, we used acetone precipitation, but I also tried chloroform methanol water, and I didn't see too much of a difference between those two. I just prefer acetone a little bit more because you can do it in one step, though there are some drawbacks to that a little bit. So, but there's a lot of published literature on how to recover proteins and precipitate them with pretty good success. And we recovered, I think, 97 to 99% recovery after precipitation. So we're fairly comfortable with that method. But the SEC is really a requirement, I should say. So I tried precipitating the sample even 3 or 4 times and still could not get rid of the triton. So the SEC actually plays a huge role in getting rid of the surfactants. We really can't do this technique without the SEC. But the benefit is obviously we see large proteoforms, which is something we can't see without a size-based separation.
Okay, last question.
Of course.
After the, you say that you get 97% recovery, that's the solid, like by a measure of the solid weight. And did you measure recovery after reconstitution?
So, yeah, so we actually ran a Bradford protein assay before precipitation and then after recovery. So it's not by the solid weight, it's by the amount of sample that was recovered. So we just ran protein assay before and after. So that's how we measured our recovery, if that makes sense.
Okay, so you did a precipitation and then 97% of that protein went back into solution.
Yes, so there's a number of really good protocols for this method. Essentially what you use is neat formic acid and then quickly dilute it into, in this case, our SEC mobile phase so that you don't get formulation of proteins. And as long as you keep it cold and dilute it, neat formic acid is an extremely good solvent for recovering intact proteins. And our results actually mimic the results that have been seen in the literature for this protocol. So they also got extremely high recovery using both chloroform methanol water and acetone precipitation for membrane proteins, as well as other classes of proteins. So there's some really good studies on that went way further than us on protein recovery after that, that you can definitely look into.
Okay, so cold formic acid. Thanks a lot.
Cold formic acid, yeah. And Yeah, if you look up, I can't remember the paper, but if you look up just solubilizing membrane proteins in cold formic acid, there's a number of studies that you can find very good protocols for.
Great, thanks. We have a couple questions in the chat. Here's from Moe. It says, great talk, Kyle. A technical question. For the azo and the cloud point membrane protein prep, what are your suggestions for working with very small amount of samples? Are there any steps that need modification? Is microgram the lowest amount you tried?
Yeah, so for, I guess one clarification for this really low sample amount that Timothy Abello worked on, that wasn't used using the cloud point. That was just using azo to extract proteins. And that's I believe 1 milligram is the lowest we tried, though I'd have to ask Timothy about that. And in terms of going even lower, I think that's more of, comes down to more engineering in terms of sample handling. So especially the work at PNNL with the the nanopores and, you know, tissue dissecting and different things like that. I think that's how you push it lower. It is kind of those automated sample processing steps at the beginning. So I think that's really the limitation from our lab's perspective was that automation in terms of decreasing any absorptivity and things that the single cell people in particular really have really refined well. So as far as I know, 1 milligram was the smallest, but I know we and others are pushing that more. And in terms of the sensitivity of the cloud point, I would say that's another slight disadvantage. It's definitely not as sensitive as our work with the photocleavable surfactant because of just kind of the nature of having that two-phase separation. And it kind of alludes to the earlier question about automating it. I think if you automate, especially the SEC and do online reverse phase, you'll dramatically increase the sensitivity, because I think where we see the most significant loss is in the offline preparations, both in terms of throughput and sensitivity. So I think multidimensional chromatography is where we'll see the sensitivity pick up on that end. I hope that answered your question.
Great. Thanks. Another question from Diliana. Hi, what is the wavelength for the azo photocleavers? If I'm working with membrane protein like GPCR rhodopsin that absorbs energy from light, do you recommend using this surfactant?
That's a good question. So the maximal wavelength is about 305 nanometers. So you need a light source kind of within this mid UV range to do surfactant degradation. In terms of working with a protein like rhodopsin, one thing that I would say is because I don't understand the application, one thing to keep in mind is that this is an anionic surfactant, so it's highly denaturing. So I wouldn't use this surfactant if I was looking to study, for example, membrane protein structure or something like that. So whether rhodopsin absorbs in that same wavelength, I believe is not going to be too relevant because it's already going to be denatured when treated with this surfactant. I think that might answer your question, but please clarify if I didn't get to that.
Great, thanks. Just a note, Victoria put up a reference in the Q&A for formic acid resolubilization.
Oh, thank you.
So we have that. Excuse my dog. FedEx guys here. But are there any other live or comments from any other folks at this point? Awesome. If not, Kyle, thank you very much. It's fantastic work on the membrane stuff. Very challenging for everyone I know, and made some great progress in trying to make this more amenable to everyday life. So thanks again, Kyle, and appreciate everyone staying on board. And September 16th, well, Frederick Lermite talking about some of his native work. So stay tuned for that. So thanks again, Kyle. Thank you, everyone, and have a great day.